Grazie al dottor.
Speaker is Dr. Louise Rodino, presente in italiano la dottoressa Rodino lavora.
Prossima studentessa, la dottoressa Dr. Rodino works with Mr. Mendel, and she's developing a new AAV to solve sarcoglycanopathies and also for muscular dystrophies and Duchenne muscular dystrophy. And I think she will talk about these new AAV vectors.
Rodino.
Good morning. I'd like to start by thanking the beta-sarcoglycanopathy group for giving me the opportunity to share our work with you today. I have Italian blood, but unfortunately I cannot speak Italian, so you'll have to deal with my English for today. So Dr. Mendel gave a very nice introduction for us on what we do in the center and for our previous experience, and it's really my job to do or lead the preclinical studies, the animal studies to support new trials. And so I have a very important job for beta-sarcoglycan, and that's to prove that it's both effective and safe in an animal model. And so I'll talk about those studies today. This is just an overview of what I'll talk about today. I'll talk about why we use AAV for gene transfer and our current preclinical studies for beta-sarcoglycan gene therapy, and that includes the outcome measures that we use and then the gene expression studies that we've done. I'll talk about the path that we're taking to get to an investigational new drug application for beta-sarcoglycan, and then I'll end with a few of the things that we're doing to optimize clinical gene transfer. We're always thinking about the next step, how to make things better, and so that includes vascular delivery and then some other models looking at the immune response. So I don't really need to introduce the limb-girdle muscular dystrophies. Dr. Mendel showed this slide previously, but what I just want to highlight is just looking at our population of LGMDs. Beta-sarcoglycan or LGMD2E is right here. It's one of the more common forms even in our group of patients. The sarcoglycans represent a large majority of the muscular LGMDs. This schematic just shows the large group of proteins located at the muscle membrane. Beta-sarcoglycan is right in the middle of the sarcoglycan complex at the membrane and really provides structural support. And what happens when you lose beta-sarcoglycan, you also lose some of the other sarcoglycans, and you also lose most of dystrophin. And you can imagine what this does to the muscle. It creates a lot of instability and creates damage to the muscle. And so really the best therapy for beta-sarcoglycan would be to restore to beta, then you would restore the rest of the sarcoglycans and also dystrophin. So that really is the approach that we are taking. Although beta-sarcoglycan or LGMD2E is very, obviously affects all of you here and is a very severe disease, we're really in an ideal time for therapy because of the previous experiments or experience with alpha-sarcoglycan and because of several advantages that beta-sarcoglycan has. We have a mouse model for the disease that really recapitulates the disease quite well. This is a problem for diseases like Duchenne, where the mouse model is quite mild and sometimes it's hard to test therapies effectively. This is not the case for beta-sarcoglycan. The model is quite severe and will help us test the gene therapy. The size of the beta-sarcoglycan gene is an advantage. It's a smaller gene. It fits nicely into our virus, and that's an advantage. We can deliver the full gene. We're now developing a large safety profile for AAV or adeno-associated virus. So each trial that we do, we accumulate safety data, we accumulate positive data, and that helps us down the road for each subsequent trial. With alpha-sarcoglycan, we determined several things, as Dr. Mendel already pointed out. One of those was using a muscle-specific promoter, and then also looking at different serotypes or different strains of the virus. So all of these studies that we did with alpha, we don't have to repeat extensively with beta, and so that helps us in both time and efforts. And then we've also spent at least 5 years developing these vascular delivery models, and this is also something that can help us in terms of time and getting to an effective treatment. So one of the first things I did several years back was to look at and compare promoters with alpha-sarcoglycan. And what we did was compare a promoter, and this is again, the promoter just tells the gene when to turn on, when to make protein. So there are promoters that work in every cell, like CMV, and then there are muscle-specific promoters. And we compared 3 different promoters to CMV, which expresses in all cells, and really found the muscle-specific promoters to sustain expression for the longest amount of time. And this was a study done in animals. And this is what led us to use this promoter in the alpha-sarcoglycan trial. So the other thing that I mentioned was that we've been developing these vascular delivery techniques. Dr. Mendel talked about one, which was the isolated or the reperfusion method. But one of the early studies that we've done, and we do this in both mice and non-human primates, is simply guide a catheter into the femoral artery. And this is a fluoroscopy image similar to an X-ray. So we insert a catheter through the femoral artery, and then we can deliver it to different arteries or different muscle groups. And this is the calf muscle that we're seeing here. But this is the model that we use in our animal studies to be able to show that we can deliver the virus through the circulation into muscle and then test it functionally. So we did this with alpha-sarcoglycan deficient mice, and this is really the proof that has now gotten us to being able to start the vascular delivery trial. What we did was delivered the virus at 2 different doses to the femoral artery of mice. And then 3 months later, we looked at their muscle. We have to sacrifice the animals. We take out the muscle. And then what we can do is stimulate the muscle and then record the force that is, or the amount of force that that muscle can achieve. And what we found, that the specifics are not important, but this bar represents the alpha-sarcoglycan deficient mice. This is normal mice. So with the largest dose, we can see that we can achieve normal function or normal physiology within the muscle. And this is important. The FDA wants us to show that we can have functional improvement for the gene therapies that we're doing. And so this is the same approach that we are going to take for beta-sarcoglycan. So how do we get to a beta-sarcoglycan gene therapy trial? So in 2000, Dr. Kevin Campbell developed an animal model for beta-sarcoglycan deficiency, and it actually it does recapitulate both the histological, the effects of the muscle, and the deficiencies in function. So we obtained this model. It was in frozen embryo form at Jackson Labs, and we've been now breeding these animals for the past several months. So the next thing we need to do was build our adeno-associated viral vector cassette. Just hit again some of the advantages of using AAV. We get high levels of transduction, especially in muscle. It's non-integrating. It persists as that episome or that little mini chromosome. Doesn't get into the genome, will not disrupt any other genes. Feasibly, we will get long-term persistence with one single injection. For this, we used, again, the same promoter, the muscle-specific promoter, and then the beta-sarcoglycan gene. We're using the human gene, even though we're delivering it to mice. We want to test the exact construct, the exact gene that will be delivered into patients, and so we're using the human gene. And so then this just gets inserted into that viral capsid, the viral delivery cassette, and then delivered. So our first experiments, we generated our vector, and then we did direct intramuscular injections into the beta-sarcoglycan deficient mice. And then we looked at just 1 month later, and these are our most recent data. We looked at the expression of the beta-sarcoglycan gene and the histology of the muscle, or the health of the muscle, and restoration of those other proteins. So these are the data that we've achieved so far. So we tested 2 different doses in the mice. And what you're seeing here is a untreated mouse limb, and then this is the beta-sarcoglycan gene expression in the treated side. So at 2 doses, you see here, at the high dose, we're achieving very good gene expression. Near 80% of the muscle fibers have the expression of the gene. This is a Western blot just showing, comparing a normal mouse with beta-sarcoglycan to both the low and high doses of the gene transfer. And you can see that we're achieving almost normal levels of gene expression. So next we looked at the histology of the muscle. As I mentioned, the beta-sarcoglycan knockout mouse does have a phenotype. You see a lot of inflammation, and you see what's known as regenerating fibers. The muscle goes through periods of degeneration and regeneration, and you can mark these fibers that are marked by this presence of what's called a centralized nuclei, this dot in the middle of each muscle fiber. And you can use that as a readout for your a benefit of your expression. So this is an untreated mouse, and then on the bottom here, this is a mouse treated by the beta-sarcoglycan gene therapy, and you can see that the muscle looks much healthier. You don't see this inflammation. You see less of these numbers of a centralized nuclei. And then this is just quantified over here. So this is a normal mouse. You would expect 100% beta-sarcoglycan gene expression and not very few of these central nuclei. And then with our therapy, we're working towards that. At the highest dose, you have 75% of muscle fibers expressed, and then very few with these central nuclei. So we're on the right track in terms of preventing the dysfunction in the muscle. Next, we looked at restoration of the other proteins in the complex. The first that we've looked at so far is alpha-sarcoglycan. You can see in the knockout mouse, you get complete loss. And in the beta-sarcoglycan treated, you restore alpha-sarcoglycan to the membrane. The same is true for dystrophin. We do see some low levels of dystrophin, but it's restored to almost wild-type levels in the treated mice. So things are looking very promising. So the next steps now are to perform vascular delivery to the mice and do as we did with alpha-sarcoglycan 3 months later, test for physiological benefit. We have not done these studies in our lab, but others have shown that we can achieve the same physiological deficits. In order to be able to show that you have function, you have to have a mouse model that has a physiological deficit. And so this has already been shown by Anderson et al. And really what we're looking at here, the assays are not all that important. We're just looking at force. So for the knockout model, you're looking at a force down here. In a normal mouse, the force is here. So we're looking to show that our therapy is at this normal or wild type level. And so that's what we'll be using as our outcome. So the current studies include continuing to breed the mice. This was one of the limiting time points for getting this started, really making the gene we did within a few weeks. We're defining the functional endpoints, defining what is the deficit in the mice. We were already doing the dose response by intramuscular injection, and now we're doing it by vascular delivery. And then doing a 3-month functional study, looking at different doses to show the minimum effective dose to improve function. So in terms of development to a clinical trial, it's a very long process, unfortunately, and we're doing it as fast as we can. But we're in the preclinical stage right here, and all the data that we're generating will be put into what's called a pre-IND package. And this is given to the FDA, which is our regulatory body, and this allows us to have a formal discussion with them. And really the idea is by the end of the year to have this discussion with the FDA and get formal guidance on what's called a toxicology study. So all of these studies, not only do we have to show it's functional in the lab and safe, but then we have a third party or an outside party do a formal toxicology study. And these are obviously critical, and we do these including at doses that are 10-fold or 10 times higher than the dose that we would use in a patient. So we have to show that there's no toxicology at a dose even higher than we would use, and these are the studies that would be following up our preclinical studies. So as always, we are optimistic and we're planning for success, and the next stage after direct intramuscular injection would be vascular delivery. And then also, we're thinking about ways to treat all patients, so we don't want to exclude the patients that have pre-immunity, and so we're developing 2 ways around that. So I'll go through this quickly. Dr. Mandel already pointed this out, but these are the studies that we've been doing in the non-human primates where we recirculate the virus by inserting catheters into both artery and vein, recirculating it. This is just an example of a marker gene called GFB, and this just shows that we get very good distribution throughout the muscle. If you quantify the number of fibers in each muscle, very good in the quad or the thigh muscle, we're achieving levels that are near 100% of the muscle fibers. So we feel very optimistic and very good about moving forward. And this is what the paradigm that we would use. It'll be done with alpha-sarcoglycan first. This will provide a very good platform for moving on to beta-sarcoglycan. So the last thing I'd like to talk about is a thing called plasmapheresis. So we mentioned that there are antibodies, or some patients develop antibodies to AAV. If you think about vaccinations that you give to your children, you develop antibodies, and that's something that happens that's less likely with AAV, as Dr. Mendel mentioned, most of us are not exposed to it, but it is still the case. So how can we get around that? And one thing we can do is perform a procedure called plasmapheresis, which is used clinically, and really this is a way of cleaning the blood, of removing any antibodies, which includes AAV. So we tried this out. We had primates or monkeys that had antibodies to the virus. We removed those antibodies, and then performed the gene transfer. And we achieved gene expression that was really equivalent to animals that did not have antibodies to the virus. So this really gives us hope for moving forward for both patients that have pre-immunity. It also gives us hope for being able to redeliver the virus if necessary. Once a patient does go through gene therapy, you will develop antibodies no matter what, and so that would potentially be a problem for readministration. But using this approach, we think we can get around that. So we're trying to anticipate all of the problems, and we're trying to develop answers before time so that we have the answer when it comes up. So I'd just like to close and say that we're working hard to make beta-sarcoglycan gene therapy reality. We're working as diligently as we can. And quickly, I just want to thank the people in the lab that are really doing this work, specifically Eric Potsguy, who's done a lot of this work and has been working hard on this. So I will take any questions. Thank you.
Thank you very much. We are open for a discussion. Some question about this presentation? Yes.
Dr. Rodino, I would like to ask you, which difference, if any, do you expect to find among the outcomes achievable with gene therapy applied to LGMD 2D and 2E?
As far as what?
Which difference you think you expect to find among the results by applying the gene therapy to these two different kind of LGMD?
We expect very similar results between the two. So far, everything is pointing to that. The same exact dose that we used with alpha achieved almost the same exact amount of expression with beta. So I think so far things are lining up to be very similar. Once we have the functional data in the mouse, we'll be able to tell that specifically, but I don't anticipate much differences between the two.
Other questions? Yes.
I'm sure you're familiar with the eagerness of patients and parents. I saw something flash by on a chart. It was too fast for me. There was a mention of a whole body delivery. So what about systemic delivery? Are you envisioning that in the far future?
That's something we're thinking about, and after we do the vascular delivery to a limb, we will test that in mice, doing a whole body systemic delivery. It's, as we mentioned, that's quite easy to do in a mouse, maybe not as easy in a patient, but we'll do that. And there's some other functional tests that we can do with the whole body to show how effective it will be when you deliver it to the whole body. So that's something that's in the pipeline, something we're developing, but a little bit further down the road, but certainly something we're thinking about.
Thank you very much for the presentation, both presentations. My question is almost related to the first one. Well, the level of delivery of viral genomes was 10 to 11 for intramuscular. You're planning to do 10 to 12, so that means one order of magnitude for the entire lower limb. What do you think? I mean, is that enough, or?
So you're talking about the first alpha-sarcoglycan
Yeah.
vascular delivery trial. So that was 10 to the 11th in a single muscle in the foot. So in the limb, we've tested this, and we do it on a per kilogram basis. So we found that we use both 2 times 10 to the 12th and 6 times 10 to the 12th in a non-human primate per kilogram. And that gave us sufficient results. So that's what we'll carry forward to the patient. It's normalized to body weight, so that's the best way to achieve results. So.
Okay. Another question? We can move to the next speaker. Yes, on you.
Thank you, Dr. Rodino.